So now I would like to call Dr. M. Sharma. He is from Stream Farmer, India, and his presentation is farming feeding the right way. So, Dr. Sharma, are you ready for presentation?
Yes, yes.
Yes, sir. Please go ahead for your presentation.
You can see my screen?
Yes, we can see.
Okay. First of all, I'd like to thank Team Conference Mind and Sonal for this seminar, and also I'd like to thank my friend Fred, the head marketing Lachance, the Shandong Longchan group. Everybody knows that I'm a shrimp farmer and doing shrimp farming for the last 28 years in Gujarat, which is the western part of India. And in this webinar, second webinar on aquaculture and marine biology, here I'm presenting feeding the right way. So because when we talk about shrimp farming aquaculture, we always talk about the quality, we talk about the quality of broodstock, we talk about pond management, we talk about management. Can I request everybody to please mute their mic because it's disturbing. Those participants, please mute their mic. So whenever we talk about shrimp farming, basically, as I told you, we talk about quality of seed, or broodstock, or the feed, the nutrition. But what as a farmers, because I've been working in shrimp farming for the last 28 years, and always every day we go to farm and attend the farm and see all what happens at the pond. So basically, very few people talk about the feeding the right way. Means what is the proper way of feeding the shrimp? Because in Gujarat or in India, we generally carry out two crops, mostly one summer and one monsoon. So every time there is a change in the climate, every time there is a change in molting patterns. So do the feeding also change? Because in also our human being, we say that what we are, what we eat. Basically the nutrition and the frequency of feeding is very, very paramount important in shrimp pond management. So before going into that, because since it's an international aquaculture webinar, let's few slides we talk about what is the present status of India in the world shrimp farming. As we know, out and out, India is one of the top shrimp producers and exporters in the world. We are neck to neck going with Ecuador in terms of production, and then you can see India was out and off and monodon country, black tiger, till 2009 when Mammalian was introduced into our India. So we can say monodon era was up to 2009, and from 2009 to 2021 right now I'm talking is a Mammalian era. This year monodon has been reverted back with SPF Moana and other SPF companies from Madagascar. They have entered the market. So shrimp farming approximately in India is close to 150,000 hectares. Shrimp production is an average 5 ton per hectare, but 80% farmers, if you see, they are below 5 hectares. So 90% we can say that our entire produce is dependent upon export and very less domestic market. So if you see shrimp production, you can see from 2010, it has grown like a tsunami, leaps and bounds. Every year we have added more than 1 lakh ton production from 2011 to you see all successive years. But if you see from 2017-18 onwards, we at the farm, whether it's in Andhra Pradesh or Gujarat, we are facing white feces, white fecal disease, white gut issues, running mortality. All issues have been piling up because of the carrying capacity of the system, and it is very clear and evident. Like 2019-20, we could see there is a drop of 20%. Here, a lot of national, international people, they are speculating that this drop might be because of the COVID and all. But to be very honest with you, from the farmer's perspective, I think COVID has done a little bit halted the production in March, April crop in 2020. But mostly the problem of the drop of 20% is mostly due to the production issues at the farm, not only because of the COVID and others. So you could see these are the major factors which has bringing down the production in India is less quality of the seed and the less survival at the farm, very slow growth and high FCR. Running mortality is the biggest damager right now. White blood, white feces, EHP. But still, if you see all these things post monsoon, we always have a WSSV as a major damager. Still 45-50% crops in India is lost because of white spot. And recently, amid COVID and post-COVID era, because of the effect to each and every aspect of life, the challenges faced is severely in the cost of production, especially in Gujarat. Because when we talk about shrimp farming, we always compare Andhra Pradesh and the rest of the India, because the cost of production in Andhra Pradesh is quite 10 to 15% is more efficient because of the 100% infrastructure available in Andhra Pradesh. The hatcheries, the feed millers, the processors, everybody is more focused in Andhra Pradesh. So the rest of India like Gujarat, Maharashtra, West Bengal has a little high cost of production. So if you see that cost of energy, when we talk about electricity, fuel cost has really increased in the last 12 months. Feed cost, fish meal, soybean, the prices have gone to sky. Labor cost has increased. Even the labor mobilization in COVID restrictions has really increased and affecting the shrimp production, the cost of shrimp production. So what is the farming feeding the right protocol? And this I am going to present whatever I have learned in the last 28 years by doing the shrimp farming. And why feed and the feed management is of paramount importance, because if you see the cost of production of the shrimp, 55-60% factor goes to the feed, the cost of the feed, and rest is all. So it is very important for a farmer to understand the importance of feed and feeding management in the pond. So the feeding management key factors, as I told you, it's 50 to 55% is the cost of production. And wrong feeding and feeding management is the mother of all problems in shrimp ponds. Because the organic load by the improper feeding management can lead to all the problems. So if you see that when we summarize the health of the pond, we talk about the good bottom, good water quality, and the health of the shrimp, the gut. So all these three factors, whether the bottom of the pond, water quality, and the gut of the shrimp, directly dependent upon the amount of feed, the amount of organic load we try to put into the pond. But we have to, the farmers need to understand the world over, especially the Asian farmers, that our entire learning, our science and our art of growing shrimp mostly focused on monodon. The moment the banami came into India, most of the farmers initially, still they couldn't, manage banami the banami way. So they used to feed banami the tiger way. That was a big blunder. And here, those who are listening to me, the scientific people, the faculty, the farmers, this is very important because banami came with the purpose of high stocking and high production. So when we talk about high stocking, percentage of feeding, quantity of the feed, and the frequency of feeding timing is of great importance in the success of shrimp farming. I will say underfeeding is still manageable, but overfeeding is the biggest crime in shrimp pond management. For example, I am giving you a very simple formula for 100,000 PL, how we should do the feeding considering that 80% survival, number of shrimp 80,000. If at 15 to 20 grams, which is considered to be at the peak of the farming, say 35 kilo feed will go into a per pond per lakh animal, for 100,000 animal. But here I would like to bring to your kind notice, many of the farmers or the nutritionists or the technicians, they hardly see how much lead is consumed by a single shrimp. I have done this experiment many times in aquarium, in the bucket at the farm, keeping shrimp 2 days, 3 days completely out of the feed, and then try to feed them some pellets. I have noticed shrimp cannot accept more than 4 to 5 pellets at a time. So if you are trying to feed them 4 times, 5 times in a day, so but if you see for the lower grade feed, in 1 kilo maximum there are 70 to 75,000 pellets. So there is no point that we are throwing 50 kg or 100 kg feed at a time, because the number of the shrimps, they will fetch the feed and it is done.
Sorry for disturbing. Sir, you can change your slide. Please change your slide.
I'm changing, no?
No, it's not visible.
Now it's there.
No, sir. Please, it's still in slide 1.
No, I will redo it. You see now? So now you can see now?
No, sir.
I think I'm able to share it. I think there is something technical problem from your end.
Yes, now it's visible. Please continue, sir.
Now you can see this slide now?
Yes, Dr. Manas.
Okay. Now you can see the example of what I am giving, like 100 PL, 100,000 PL formula. You can see the slide now? Sonal?
Yes, Dr. Manas. Now you can go ahead.
So for example, when I was talking about, first of all, sorry for the technical glitch, because we are into completely depend upon the internet and its speed. So let's go back to the slide of feeding for 100,000 PL. So survival, if you consider 80% and number of shrimp 80,000. So total feed during the size of 15 to 20 grams. So we say 35 kilo goes to the pond. But as I told you, many people hardly notice how much pellet is eaten by a shrimp at one go. So I have noticed by doing these experiments in aquarium and in the bucket at the farm, keeping the shrimp off the feed for 1 or 2 days. But what I have noticed, maximum shrimp will consume only 4 to 5 pellet at a time. So total pellet required per feed, if you calculate, it is like 360,000 pellets are required to feed 80,000 shrimp at the ABW 15 to 20 gram. So the number of pellet per kg is 70 to 75,000. If you really count what the lower one number, if you count it in 1 gram or 100 gram, you will see that not more than 70,000 pellets, 70,000 pellets falls for 1 kg. So feed per meal, maximum, then you need to, generally what we follow is that 3 times feeding or 4 times feeding in shrimp farm. But as I noticed, because it's a high density stocking and anami, you need to increase the frequency of feeding as the quantity of the feed increases in the pond. There is no point like throwing 50 kilo, 60 kilo at one meal, because it is going to be severely wasted at the bottom. So you could see feeding which has originated in our country, like diet feeding, whether it is good. Sonal, you tell me if the slides are not moving. Diet feeding is good, or you can see the diet feeding which is better, because we are trying to throw very much inside the pond, from the margin of the pond. Boat feeding it has proven to be very excellent. You can see one of the latest technique in India and the world over is auto feeder, which is going to be very excellent in managing the feed 24/7 into the shrimp pond. So now you can see all the slides. Hello?
Yes, now we can see diet feeding good.
Okay. But I think it's going very slow.
Yes, maybe.
Now, but you can see the now auto feeder slide.
No.
I think Sonal, others are visible or it is only not visible to you?
Now it's visible. Now it's changing. It's taking some time.
Now you can see the solar autofeeder.
Yes, Dr. Manoj.
So I have given here manual feeding and its efficiency. Now you can see this Sonal, please let me know. Manual feeding, the slide. Yes, sir. Please, I am little confused because half people are seeing my slide. So please respond when I am saying. Pond size, if you see 100 meter by 100 meter, that is called pond. So if you see that manual feeding, the feed through distance cannot be more than 5 meter. So if area cover into a pond, technically when one labor goes and he throws the feed all from the dike, so maximum he covers only 1000 meters. That is 10% of the pond only he is throwing the feed. So imagine when the shrimps are all over the pond and your labor and your manual feeding is only going into the 10% of the pond. So the number of pellets and the numbers of feed per meter square is exorbitantly high. So the number of pellets per meter square per kg is 70. Imagine the kind of feed waste or feed mixing with the water. You can imagine the waste which will go to the pond. So I have given here now comparison. This is a very important slide. So Sonal, it is visible to know? Comparison between manual and auto feeding. So if you see the feed through distance, radius is 10 meter in auto feeder. Effective feeding area is 314 meter. And whereas the manual is 1000 meter only. But the quantity of feed dispensed in auto feeder is 200 grams per every throw. And in manual it is 25 kilo plus when you throw. So number of pellet dispenses in auto feeder per throw is 13,000. Whereas you throw the pellets at the level of 25 kg is more than 17-18 lakh pellet. So number of pellets available for shrimp in auto feeder is just 40, whereas the pellets available through your manual is 1750. So the density of shrimp is considered 30 piece. Imagine in auto feeder, one shrimp will get one pellet at a time, whereas in manual feeding for one shrimp, 58 pellets have been thrown and they are vested in the pond. That's where when we see the shrimp farming after 60 days, the water quality gets deteriorated, the water quality becomes thick, the bottom becomes bad because as the peak of the feeding in shrimp farming, especially in Manami, attains at 60 DOC, so there that becomes the biggest blunder in feed management of the pond, especially feeding. That's what I'm saying, feeding right way is of paramount importance. So do not feed Manami the tiger way. This chart is very, very clear. If you see, if your biomass is 1 ton, 1000 kilo, and when you see ABW3, you need 70 kilo. If your ABW is 4, you need 60 kilo. Likewise, when you have a 1 ton biomass of 30 gram size, you need only 15 kilo feed to be given for the biomass. So that is very, very, very important for farmers to understand. Do not feed like monodon to Manami. So here I have given you a very simple graph that if now right now also we are reverting back to monodon. For monodon, when you have a 30 gram shrimp, you need minimum 20-22 kilo feed. Whereas in Manami.
Hello, Dr. Manoj.
Yes.
Sorry to disturb you again. Sir, we are staying in solar auto feeder slide.
I think, ma'am, this is not happening from my side, and I think definitely there is something wrong at your end because 3 times I have changed it.
Sir, other people are also commenting slides are not changing.
So ma'am, you can start from your side because I already gave you the presentation.
Okay, then we will presenting from our side.
Do it fast because already I will stop sharing from my side.
Okay, sir. Please wait for 5 minutes. We will presenting from our side.
Sunil, you can share the screen, please. Yes, yes, from this. Yes, this one. You can make it full screen. Sunil, make it full screen. So in manual feeding in the pond is 25 kilo plus. So number of pellet dispenses is 13,000, whereas 17.5 lakh pellets have been grown with 25 kilo feed manually. So density of the shrimp is considered 30 piece with auto feeder also, with manual feeding also. So the number of pellets available for the shrimp to pick up at one time is only one. Whereas in manual feeding, you see 58 numbers of pellets are available for the shrimp to come and eat. So imagine the kind of waste it happens in improper feeding at the pond level, and most of the farmers, they are not aware of that. They just learn what is given by the company, feed company, the suggestions and the feed administration protocol. You feed 2 times, you feed 4 times. But for everybody's surprise, at my farm, where I don't have
automated gene expression, were also affected by dietary fish meal or levels. And for other problem, maybe the immune response. We have done a lot of parameters. For example, we have done some ER stress related gene expression, and we also done some histology changes. We could find that the low fish meal diet impaired the health of shrimp. So do we have our feed additive could improve the lipid metabolism and the immune response of shrimp? I think the bile acids may be a good choice, because bile acids, as we know, it plays an important role in the digestion and absorption, especially for the lipid digestion. So it could improve the lipid metabolism of the shrimp, and the bile acids also serves as a critical signal molecule in regulating immune response, and it is also a major regulator of the gut microbiota. So we think bile acids also can improve the immune response of the shrimp. Actually, in the last year, some researchers have reviewed the bile acids metabolism in fish, and they found that dietary fish meal alternatives often disturb bile acid status in fish, resulting in either increased excretion or decreased intestinal reabsorption, and decreased bile acid synthesis. And this situation may be mitigated by the processing of fish meal alternatives, as well as the dietary inclusion of some bile acids. And although the bile acid is traditionally regarded as unsynthetic in crustacean, and now a few recent studies have revealed the critical role of bile acids in the diet of shrimp, especially this article published in Aquaculture Nutrition. It's published in this year. The authors have found the special role of bile acids on the growth, performance, lipid metabolism, non-specific immunity, and intestinal microbiota of wet shrimp. And in this study, we used the CDC, or primary bile acids, which widely used in human and animal studies. And we want to evaluate whether bile acids could elevate the negative effects brought by low fish meal diet. So then I will present my first experiment. Dietary supplementation of CDCA improved the growth performance, immune response, and the intestinal health of juvenile Penaeus monodon with a low fish meal diet. In this experiment, 3 data was formulated. A high fish meal diet with 25% fish meal and a low fish meal diet with about 15% fish meal, and the fish meal is replaced by the soy protein concentrate, and also a low fish meal diet supplemented with 0.1% of CDCA. And in the low fish meal diet, some amino acids and minerals and vitamins were added in the low fish meal diet. And in this study, the shrimp used is about 0.3 gram, and there are about 30 shrimp in one tank, and each diet was sent to 4 replicas. And the experiment is lasted for about 8 weeks. And first we can see the growth performance. The shrimp feed the low fish meal diet, their feed body weight, weight gain, and feed efficiency are all lower than those in high fish meal group. And the low fish meal diet supplemented with CDC showed no difference with other 2 groups. It could say that the CDC partially improved the growth performance of shrimp. And we also measured the lipid metabolism related gene expression. And we can say that the dietary supplementation of CDC also improved the lipid metabolism of the shrimp. And we also detected the gene expression of immune response in Hepatopancreas. We can see that the expression of SOD, CAT, and radish, their expression all enhanced by the supplementation of CDCA. And similar results were found in the immune response in the intestine. Also, we can see that SOD and CAT, XBP1, and radish, their expression are affected by the dietary supplementation of CDCA. We also measure the histology of the intestine and hepatopancreas. We can see that the intestinal fold heat was lower in low fish meal group, and we can also say that the ER swollen was found in the low fish meal group. So could you say the slide change? Could you say that? Sorry, could you say the changes of slide?
Yes, we can see.
Okay, no problem. And we can see that the dietary supplementation of CDCA improved the intestinal health of the shrimp. And we also measured the intestinal microbiota of the Penaeus monodon. And we can see that the diversity and the richness of the intestinal microbiota was decreased in the low fish meal group. And the dietary supplementation of CTC elevated this situation. And further analysis showed that there are clear differences in intestinal microbiota of shrimp feed the different diets. And in this figure is the significant feature of pheromone. So we can see that the Proteobacteria was lower in the group of supplemented with CDCA. And here is the significant feature of top 50 general. Dietary supplementation of CDCA could decrease the harmful bacteria abundance, especially the Weber. And we have done some further analysis of the intestinal microbiota, and from this figure we can see that the Weber and the Proteobacteria will also enriched in the low fish meal group. And the conclusion for this experiment, the first will be low dietary fish meal decreased the weight gain, impaired the immune ability, induced intestinal damage, and disturbed intestinal microbiota of Pineus monodon. Dietary supplementation of 0.1% CDCA in the low fish meal diet enhanced the antioxidative ability, elevated the ER stress, improved the microbiota diversity, and decreased the harmful bacterial abundance of Pineus monodon. And then I will show the second experiment. This experiment is dietary supplementation of CDCA on little Pineus vaname, feed the diet with fish meal replaced by Clostridium auxentium protein. And this protein is a kind of bacterial protein which can produce the alcohol. And for this kind of protein, it has very high protein content with about 85% and extremely low lipid content. And in this experiment, 5 diets were formulated. Also a high fish meal diet, a low fish meal diet, and 3 diets with different dosage of CDCA. And actually, this experiment is just finished in the early of this month. So until now, we only have the data of growth performance. And from this table, we can see that dietary supplementation of 300 and 600 milligram CDCA partially improved the growth of shrimp. They have no difference with the high fish meal group. And what we are working on now, we are doing the transcriptomics change induced by low fish meal and CDCA. And we also done the intestinal microbiota changes induced by low fish meal and CDCA. And we also detected the intestinal health. And what's our future plan? We just started a new experiment with Shandong Longchang, and we want to evaluate the commercial bioacids products in the shrimp, and we also want to compare the difference between the commercial bioacids and the pure bioacids such as CDCA and the UDCA. And we also want to study the interactions between dietary lipids and the bioacids. And maybe we can share our future result in the next meeting. And thanks for your attention. Thanks, everyone.
Thanks. So, very good morning to Sonali and all the audience. This is Ben from Shandong Lachance. Before start my topic, I would like to say thanks to organizer and all the speakers and my team members. Okay, so for the slide ready. So for my topic, I will highlight two parts. First is for the health level of aquatic animals, which is very, very important. The second part is for our solution. If the liver damage, how can we do? What can we do? Okay, because we attend so many international conferences, but seldom report is on. Seldom reports are on the importance of liver health. Today, I will share some information on the health liver. So please go to next. So my topic will be covering the following four parts. First is global aquaculture introduction. I think yesterday a few speakers, they have shared the information already. And the second part is our solution, which is on bile acid. And the last part is application for bile acid in aquatic animals. Next. Okay, next. So for the major species produced in water aquaculture, the finfish is the higher percentage. The top three is grass carp, silver carp, and neuroticus, which is the tilapia, almost 30+% of total global aquaculture production. And for the crustacean, the top one is white shrimp, which is the venomous, and the second is crayfish. In Chinese we call 小龙虾. The third one is mud crab in China. Okay, next. So in China, top 5 countries, which is China, India, Indonesia, Vietnam, Bangladesh. China, we can contribute 57.93% of global production of aquaculture based on FAO in 2020 to 2021. And the second place is India, which can produce about 7 million tons. Indonesia is 5 million tons. China area can produce, the aquaculture, not wild caught. Only aquaculture, we can produce 47.559 million tons per annual year. And Bangladesh also contribute around 2.4 million. Please move to next. So that means that Asia is still the biggest producer in the world. Okay, we can contribute about 88%. And please move. Okay, back to 5. Back to 6. So Nelly, please back to PPT 6.
Yes.
That's 6. This is.
Is this the correct one?
1.3. Back to 1.3.
This one?
No, back. Yes, this one.
Okay,
I think for the farmers, for the aquaculture industry, we always highlight the health of intestine, but seldom report or seldom farmers, they highlight the industry, they highlight the liver health. So today I will share some information on the importance of health liver. So from the photo we can see the first is the health liver. I cannot see. Okay. We can see the picture one is the health liver is red, and the second and the third one is yellow and white liver, which is very unhealthy. And for the bacteria, virus, parasites, many products, many companies are doing such kinds of products which can treat. But for the fatty liver, not that many companies. Okay, next. And we have do a lot of investigation on the fatty liver. The first is high fat. As we all know now, the fat is the diet, the high fat diet, the fat content is keep increasing from 5 to 6, 7, 8, even 15%. And the fish cannot digest the fat in the diet. Then the store in the body and finally damage the liver. If the liver unhealthy, will affect FCR will increase, FCR will be increased, and the growth rate will be lower, and the mortality. Then finally cause the lower profit. Okay, so next one. And for the shrimp, the similar. After in the past 5 years, we do some investigation on the hepatopancreas of shrimp. We found the hepatopancreas is very important for the production. Okay, for the WSSV, TSV, IHHNV, EMS, EHP, we have a lot of papers, we have a lot of published research we can refer to. We have the solution, but for the hepatopancreas, the health of hepatopancreas, so far not that many data. So we do, back to 2 years, we start this project. We publish a few papers on the hepatopancreas. Later I can share to you. Okay, next. Hepatopancreas of shrimp is the most important organ. I call it engine of shrimp. Okay, the function like secrete digestive enzyme, absorbing nutrients, transport the nutrients from this organ to the other organs, and the storage of energy like the lipid. When we see the hepatopancreas with the wet membrane, which is lipid, fully covered with lipid, white color is healthy hepatopancreas. All in all, hepatopancreas plays important roles during the growth reproduction process in shrimp. Okay, so far we found, I think some farmers also have the similar experience, which is WFD, white feces disease. And yesterday, Dr. Manuji also mentioned this is quite common in Indian shrimp farming, in China, in Asia, and even in Latin America also this happen, white feces syndrome. So actually the white feces not the real feces, actually is the absolute cells of hepatopancreas and the intestine, which means after disease break out, they damage the hepatopancreas, finally got the white feces. Okay, please move to next. And our solution is bile acid. Later I will introduce the functions of bile acid. Next one. What is bile acid? Bile acid is a mean active components of bile. Is synthesized in the liver cells using cholesterol as a precursor, and also can be artificially synthesized too. Okay, the first function is protecting liver and gallbladder. In the shrimp and the crab, the crustacean will protect hepatopancreas. The second is promoting lipid digestion, absorption, and metabolism of lipid. The red food too is for human being, which is a cholic acid capsule. CDCA, later I will do some explanation. Next. So composition of bile acid. Actually in the nature, only less than 10% is free bile acid. And more than 90% is conjugated bile acid. The free bile acid include cholic acid, deoxycholic acid, quinodeoxycholic acid and lysocholic acid. And conjugated acid bile acid have GCA, taurocolic acid, which is a free bile acid combined with glycine and taurine, and like GCDCA, TDCDCA. Okay, next. So what's the functions of bile acid? Next. So bile acid have three functions, which is emulsification similar to emulsifier, and increase the lipid activity and promote the absorption of lipids. The first is emulsification. Bile acid. This is a bile acid, cholic acid structure, and it has two sides, which is a hydrophilic and hydrophobic. Later I will do. This is a fat globule. Okay, bile acid combined with the fat globule cause the big particles break down into the small particles surrounding by the bile salt, then can dissolve in the water. Okay, this is a emulsification. But remember, emulsification is not digestion. They only break the big lipid particle into small droplets, which is increase the surface area and make it available for digestion by lipids. Because as we all know, lipids cannot access the inside of access the inside of lipid droplet. Okay, that's why we need the emulsifier to break into. Okay, this function is similar emulsifier, but for the bile acid, it has two more functions. Next. Okay, the second function is digestion. This is triglyceride and lipids, bile salt. Then triglyceride surrounding by bile salt. Okay, bile salt can break the lipid. This is the lipids, the structure. Okay, expose the catalytic groups, form the hydrophobic channels, then digest the lipid. Okay, this is a function 2, which is the emulsifier doesn't have. Okay, next one. And I think this research back to 1982 from Japan, they have do a lot of research and publication on the eels on tilapia. Yes, this tilapia is 2017, published by our companies, which is SCI papers, in the frog, in the grass carp, in the perch and turbot. Plenty of evidence which bile acid can increasing lipase activity. Okay, move to the next. Okay, the last function is absorption. Okay, they will form, this is the emulsification just now I mentioned, to the small particles and increase the lipase activity. The last one is absorption, which is most important. Okay, both fatty acid and bile salt will form the mast cells, which can dissolve in the water. Once dissolved in the water, can close to the absolute cell to absorption. Okay, go to the blood, go to the capillary, back the liver. This is if the particle cannot digest. If not mast cells, the particle too big, okay, they will store in the body. And when we open the fish, we will see a lot of white material, which is a lipid, which is a fat in the belly. Okay, so this is very important. So move to next. Also have some research on mammalian cell. Yesterday, Prof. here also mentioned similar, bile acid interact with the gut microflora. Okay, this is for mammalian. I think from mass, it's very mature technology, but in the aquaculture so far, I didn't find any papers on this. This is a bile pool. Okay, this is a gut microflora. They will interact together then as a regulator to promote the lipid digestion. The theory part is very complicated. Different animals, they have a different pathway. Okay, next. And I just do a quick summary. The first, bile acid can promote the absorption of fat and fat-soluble vitamins. Fat-soluble vitamin also stimulate intestine to secrete digestive juice, peristalsis, and so on. And the second is free bile acid. Just now I mentioned the 4 types of free bile acid, which can inhibit the growth of a variety of intestinal bacteria, and also can maintain the barrier function of mucosal and the stability of intestine environment. This is also very important function. Next. So the last part I will share some application in aquatic animals, include some papers, scientific papers, and also in the other countries, the field case. Next. Okay, this paper is on replacement of soybean meal, replacement of fish meal with soybean meal. Okay. CB, we use the CB. The fish meal contains 50%, but once we add 53% soybean meal and plus 5% fish meal. Okay, after 90 days, we can see the result. The body weight from the fish meal and the soybean meal drop almost 20%. So the fish meal gets a good growth rate. And the feed efficiency also dropped around 18%. Daily feed intake increased almost 10%. So that means when we replace the fish meal with the soybean meal, it does affect the growth of the fish. Okay, finally we also test the cholesterol in the blood, dropped around 28%. And later, next. And we do another is on tilapia, which is promote the feed efficiency, growth rate, and also increase the SGR. This is for the neurotakers. We have 5 treatments, one control, and 4 treatment, which is 50 grams per ton feed. That means we add 50 grams bile acid in 1 ton feed, 150 for 450 and 1.35 kilo per ton feed. Finally, we found 150 grams per ton feed group got the best result in terms of final body weight, weight gain, and feed efficiency, also HSI. Okay, the final body weight increase from 74 to 82.5, which is around 10%. Weight gain also similar result. Feed efficiency improve 15%. Okay, this is a good news. I think because just now I mentioned bile acid can promote the digestion, absorption of fat. This is one of the reasons. Okay, survival rate, because this is a neurotakers, is hardy fish, only increase 2.5%, but it is acceptable. Okay, move next. We also investigate some lipid in the muscle, also in the whole fish body. Let's say 150 gram treatment. The the crude lipid in the body is dropped. How many percent? Around 20%. And in the liver, in the muscle and the liver, we found the similar result. So that means the bile acid does promote the digestion of lipid in the fish body, not only in the feed. Okay, next. And for the lipids, increase the activity of lipids around 60%. In the intestine and the liver is a similar result. If you are interested, we can share these papers to all of you. Okay, next. So from last paper, we can see 150 gram bile acid in 1 ton tilapia feed. It does help to increase the survival, promote the production, and also increasing the lipid activity. Okay, this is another paper I think done from Taiwan researchers, professors. If you will add some bile acid in the shrimp feed, it will reduce the virulence of parahemolyticus, the toxic of Vibrio. So this is very important and useful for the farmers. Okay, because AHPND is very common in Asia. In the most of the shrimp farmer, they are facing the AHPND. Okay, if this bile acid can solve this problem, I think the market is huge. Okay, and our production, shrimp production, will be increased a lot. Now we only have all over the world. We only have 5 million tons of white meat production, metric tons. China contribute around 120 to 150 million tons. Okay, so you can search this paper. Okay, next. And this is recently we published in SCI papers done by our company and the China Ocean University of China. We have the 4 papers, which is replace of cholesterol in the feed. Because we know cholesterol is very expensive. 1 kg is around 700 Chinese yuan, which is about 100 plus USD. Okay, we are trying to replace this one. So next. Okay, we designed 4 9 treatment. The first one is 0.15, which is 0.15% replacement result bile acid. Okay, diet 2, 3 we add bile acid, and one diet 1, 4, 7 without any bile acid. We only add cholesterol. The rest 6 groups, we add different level of bile acid. Later we can see the result. From the diet 4 and 5, we compare because we found the weight gain, the final weight, this is the highest. That's why we choose this diet 4 and 5, which means replace 0.2% cholesterol using bile acid. So that means the feed cost will be dropped around $5 per ton feed. And finally we found, okay, the final weight gain, survival rate, FCR drop, the SGR increase, and HSI also drop a bit. Okay, next. Okay, we also test protein, lipids, amylase, and SOD. These 4 parameters all increase compared to the control, even higher than diet 4. That means when we replace cholesterol, we add some bile acid. it will help. It will help the shrimp to get the higher survival rate, higher production. Okay, 0.2% replacement and add 200 gram bile acid is the best treatment. Okay, next. And after that, we did dissect the shrimp, get the Hepatopancreas. So ADG, which is a result bile acid. From B, C, E, F, H, I, we add the bile acid. Okay, as we all know, Hepatopancreas include 4 types of cells, which is B, R, E, F. Of course, some researchers mentioned is 5, but not much different. So from A and D, we can see the cells, quite a number of B cell and R cell degeneration and disappeared. Of course, the tubular and epithelial cell also rupture. Okay, but for the B, C, E, F, G, H, I, it's very clear. We can see a lot of B cells, R cells, and E cells and F cells, which means bile acid does help protecting the Hepatopancreas of shrimp. Okay. Please move to next. So of course, our product is bile acid. The main component is bile acid, but the commercial brand is Renew 2. Now we have export to 45 countries and also using in tilapia, eel, perch, sea bass, grouper, shrimp, frog, and so on. Of course, we are also using poultry, using ruminant, and the other culture species because bile acid produced by the liver of the animals. They have but sometimes from animal itself is not enough. That's why we're adding extra bile acid. Please to next. Of course, this is a case study because this is from India. Now we get very, very good result. Also, the Dr. Manuji, his farm get good production of shrimp. Okay, this is from the other farm. Okay, next. And this is from Malaysia. Is a high density intensive shrimp farming, which is 200 per square meters. In the run tank. Okay, they add 2 to 3 grams of bile acid per kilo feed. Okay, next. And this is from Indonesia. The happy fish. Yeah, we did short the growth around 10%. Before they need around 90 to 100 days. But now they only need 80 to 90 days to get the same weight of the shrimp, the final weight. Okay, next. This is from Thailand. I'm not sure whether we have the speakers from Thailand during this webinar. And they culture the vanami and also tiger prawn. You see, the healthy, very nice tiger prawn. Next, from avocado, because now we know avocado is every year they can produce almost 0.7 million tons, which is the second or third place in the world. Okay, the density is very, very low, around from 12 to 25 per square meter, but the production is very consistent. So after using our Renew 2, the production is the same density and the same culture period, the production increased 5 to 10%. Okay, next. And of course, the second, maybe if Aquado is second, Vietnam is third. If Vietnam is third, is second, then Aquado is second. Okay, this is after using 1 month, we can see clear hepatopancreas, the thick intestine, the shrimp, and this culture density also very high. They lime the pond with HTP PE membrane and the cover with the net. Next. So come to the conclusion. First, the bile acid protect the liver and all hepatopancreas and the gallbladder. This is true because after 17 years trial, we approved this one. In China, we have also using a lot. And second is promote the fat digestion of lipid from the fish body and the fish feed. Just now we see we test the lipid in the muscle, we test the lipid in the liver or hepatopancreas is decreased compared to control, which also reduce the body fat deposition. The third is bile acid in the artificial feed can promote the growth 5 to 10%. Okay, and increase the survival rate of the fish or shrimp, depending on your technician. Now we found for the hardy fish maybe a bit lower, but for the shrimp, at least 5%. Of course, because now we use a lot of plant protein to replace the fish meal in the shrimp feed, in the fish feed. Okay, we found if you add too much plant protein, after 2 months, your shrimp most probably got wet feces. Because there is evidence, we also conduct such kinds of project to approve. If add too much plant protein, will damage the cells of hepatopancreas. After 2 months, definitely, if the environment become worse and the weather or something happen, your shrimp will got wet feces disease. Okay, and if now we trying to do another project which is on also on replacement of cholesterol, and also we will investigate the health of hepatopancreas and the liver of fish. Okay. So next. Thanks. I think this is all my topic. Thanks, Sonali and audience. Any questions?
Thank you for your excellent presentation.